This recording is going to cover specifically the process of gamete production in the male referred to as spermatogenesis. Now, in the male, spermatogenesis is actually going to occur within the testes, which you find located right here. And just a reminder that that process of spermatogenesis is important, that the temperature is kept a couple degrees less than body temperature. Therefore, that's why the testes are located in the scrotum outside of the body. And review the significance of both cremaster muscle and the dartos muscle in keeping the temperature optimal for spermatogenesis, as well as this pinniform plexus. So I'm just reminding you from previous recordings about that. Now, within the testes specifically, we should say that spermatogenesis occurs within the seminiferous tubules, which you see here. And if we kind of look at a slide of it, you see all these seminiferous tubules here. Here's one here, and what you're seeing is sperm cells at various stages of development. You'd have to look really carefully, but remember, this is a slide, so everything's not going to be perfect. Things get washed away. But the spermatozoa, the ones with the tails, would actually be located closer here, and the stem cells would be located out here. So we'll be going through each component of that. But also a reminder out here, the Leydig cells. Those Leydig cells produce testosterone, which are actually important in spermatogenesis. Now, what I want to do first, though, is talk about the function of Sertoli cells. When I was a student, we kind of called the Sertoli cell nurse cells or sustentacular cells. I want you to call them Sertoli cells. The Sertoli cells, you kind of see all the cytoplasm, the Sertoli cells right out here. They're going to be kind of surrounding all those sperm cells in various stages of development. Now, they have a lot of functions. They're very, very important cells. Besides producing various factors and nutrients that aid in the whole development of the sperm or spermatogenesis, they're going to do other things. So I want to go through some of the other functions. So one of the functions that they do is they produce a hormone called inhibin. So remember, gonadotropin releasing hormone, which is produced by the hypothalamus, targets the anterior pituitary here, and it stimulates the secretion of the gonadotropins, FSH and LH. Well, LH targets these Leydig cells, which you see here. And those Leydig cells produce testosterone, which will be important in spermatogenesis. Now, FSH specifically targets the Sertoli cell. Please write that down because that's kind of important, that the receptor for follicle stimulating hormone is on the Sertoli cell. And it's kind of just like what it sounds, follicle stimulating hormone. So the Sertoli cells do a very important role in stimulating the development of the follicle, in this case, the sperm. So the FSH stimulates the Sertoli cells to do all the stuff that it does. And one of the things it will do is it will produce a hormone called inhibin, which inhibits FSH. So inhibin inhibits FSH. Now the question is why, and it's really only FSH, it doesn't inhibit LH, is if the Sertoli cells want to be able to manage a certain number of or sperm cells. It doesn't want to become overwhelmed. And so to try to keep things in check so they don't get overwhelmed, so they can do their job properly, is it releases inhibin, which will kind of decrease FSH so we don't keep producing too many sperm cells that it can't handle. So that's one of the things that inhibin does. And obviously, as I mentioned, the Sertoli cell produces inhibin. Another thing that it does is it produces something called Mullerian inhibiting substance. This is released during fetal life. So during fetal life, what this will do is it inhibits the Mullerian ducts in the male embryo, and what would happen is the Mullerian ducts in the female it becomes like the uterus and the fallopian tubes. So we don't need that in the male. So it will inhibit those Mullerian ducts. The Wolffian ducts, just so you know, the Wolffian ducts that you see written here, is they'll die if there's no male hormones present. So in the female, when you don't have those male hormones, the Wolffian ducts regress. But in the male, the Sertoli cells are going to produce this Mullerian inhibiting substance, which helps to cause the Mullerian ducts to regress because we don't need those in the male. Now, another thing that it does, I've got to go back here, is that the Sertoli cells produce something called androgen binding protein. Testosterone is an androgen. And for normal spermatogenesis, you need very high concentrations of testosterone within those seminiferous tubules. So the FSH induces the Sertoli cells to make the androgen binding protein, which would be abbreviated here as ABP. That androgen binding protein binds to testosterone and prevents it from leaving. So it keeps the testosterone concentrations extremely high, so it's needed for proper spermatogenesis. Now, one more thing that those Sertoli cells do, and you can kind of maybe see it here, is you'll notice there is endothelial cells here, and here it says lumen of capillary, which is blood. Well, we have to maintain remember learning about a blood-brain barrier? Well, we have what kind of, I'm going to use air quotes, a blood-testis barrier, or some people refer to it as a Sertoli cell barrier. It's an immunologic barrier. Okay, so I'm going to write that out for you. Immunologic barrier. It provides a specialized microenvironment for those developing sperm cells, because what can happen is immune cells that are floating around in here will recognize those sperm cells as being not part of that person, part of that male. They're not going to be considered self. And so what will happen, those immune cells will destroy those developing sperm cells. So the Sertoli cells are going to provide that specialized environment, a kind of like immunologic barrier, or provides immunologic privilege to those sperm cells so they can develop properly. So those are like pretty much what the Sertoli cells do. They provide a pretty good, pretty important function. And I always ask questions about Sertoli cells on the exams, just like you know. Here, what we're going to see here is the process of spermatogenesis. Remember, spermatogenesis is a result of meiotic divisions, and meiosis is a biological process by which diploid cells undergo cell division to form haploid gametes. So in the male, spermatogenesis begins at puberty. Please write that down. So it starts at puberty and then continues throughout their lifetime. Now, in the female, oogenesis starts to begin during fetal development, but in the male, spermatogenesis does not begin until puberty. What happens is, on those outsides of those seminiferous tubules, you start with spermatogonium. So you have spermatogonium here, which are diploid, and that is what's represented here, is a spermatogonium, and it's diploid. And remember, a diploid refers to the number of chromosome sets the cells contain. So if it's diploid, it's got 2 complete sets. Well, what will happen is that spermatogonium will undergo mitotic division. And here's one of the spermatogonium that results from it, and that decides to hang back. I'll just wait here for some other time. Well, the other one, the other daughter cell, will decide, okay, I want to become a primary spermatocyte. So this one here is a primary spermatocyte, which you see here, that is still diploid. It's going to start kind of doing some undergoing meiosis, and this still is right here, still a primary spermatocyte. When it completes the first meiotic division, or meiosis 1, you have 2 secondary spermatocytes, which you have written here. Okay, so you have 2 secondary spermatocytes, which are haploid. So you see the ends instead of 2 ends, so they're haploid. After the second meiotic division, you're going to end up with a total of 4 spermatids, which you see here. So this one divides, you get 2. This one divides, you get 2. So you get a total of 4 spermatids, which are all haploid. Now the spermatids don't look like sperm because they don't. What will happen as a result of a process called spermiogenesis, which you see written here, what you'll see is the shedding of excess cytoplasm, the development of the tail, and it's going to start to look like what people would envision as a spermatogonium. Or sorry, not spermatogonium, a spermatozoa. So these, you end up with 4 viable haploid spermatozoa. So let's look at the structure of the spermatozoa. Now I have to mention again that this whole process is taking place within the seminiferous tubules. And this whole process takes about 64 days, which is kind of amazing. But it does take about 64 days for this whole process to occur. But it's continuous. It's continuous. Once a guy hits puberty, it is continuous. Now let's look at that structure of the spermatozoa. This top part here is the head. There's a lot going on in the head. So the head contains 23 chromosomes. So that's haploid, because diploid would be 46. And it's covered by a structure called an acrosomal cap. So I'm going to make the cap gold or yellow. So we'll say the cap is like right out here. Okay. So that cap contains enzymes that help to penetrate the oocyte. So that's needed to get through the layers of cells that are surrounding that egg in order for fertilization to take place. But in deep within the head, also what you're going to see, because I said it contains the 23 chromosomes, so I'm just going to write in here, this is the DNA. This is where the chromosomes are. Okay, so the head has the chromosomes, plus it's covered by that acrosomal cap. Now the center part here is referred to as the midpiece. Now this actually here is called the neck. This is the midpiece. Now the midpiece contains a lot of mitochondria. Mitochondria produce energy, produce ATP. That's needed for energy for movement of the sperm's tail, which you see here, as it travels through the female reproductive tract on the way to fertilizing the egg. Now that tail is a flagellum. So this is the tail, and it actually is a flagellum that provides locomotion. So it moves, and it gets that energy from the mitochondria producing the ATP. Now, as a little quick reminder, is that energy that is the fuel source that's used for production of the ATP is actually fructose. And try to remember which of the glands in the male secrete fructose. So I'm just going to see if you know that. I'm not going to answer it. You need to know that. So this is just the end of spermatogenesis. Another recording will cover oogenesis.